What's up, Hippie Bio Penguins? Coming at you again in the second time for the same day. But we are doing our FRQ Friday. It's Friday. Yeah, I don't have anything, sorry. So we're going to do 3 questions today. So the 3 we're going to do, we'll do 2016 number 7, 2015 number 4, 2019 number 3. And again, if you have questions, you can just put them down into the chat, because I can see the chat as I'm working, so I can easily see your questions, so I can answer any questions we have about the free responses as we go. So without further ado, let's get started. And they're all short FRQs, but they have kind of longer prompts going into them, so just kind of bear with it. This is, if you go to AP Central, you can pull up all the questions. So AP Central has all the questions organized by year. You can also just go to Google and you can type in the year and the number, and you can easily pull up the question. You can also pull up scoring guidelines. And then I post a copy of this PowerPoint onto my Weebly afterwards. So apbiopenguins.weebly.com. And I may have actually already uploaded the Weebly in preparation for tonight. So, the PowerPoint may already be up there. So you might be able to grab that if you're wanting to look at it as I go. So first question we have is 2018 number 4. So we have this common bug. Bed bug is a species of insect that is increasingly resistant to insecticides. Bed bugs possess several genes suspected to contribute to the resistance, including P450, ABC8, and CPS. To investigate the role of these genes in insecticide resistance, the researchers deleted one or more of these genes in different strains of bed bugs, as indicated in Figure 1. Treated the strains with the insecticide beta something. Each strain was genetically identical except for the deleted gene and was equally fit in the absence of that medicine or that chemical. The percent survival of each strain is following the treatment is shown in that figure. So we have this figure down here, but that's kind of small. That's like a nice big. Okay. So here we see the percent survival. So you can see that the most survived in 1, and then we had a high number of survival in this IV. Also kind of look at the error bars. Notice that the shorter the error bar, the more concise the data is, and more accurate it is. So you want there to be less variability in the data versus this one has more variability. And then you can see down here whether they actually have it or not. So pluses are indicating that they do have the gene. A negative indicates that it doesn't have that gene. And so the first question we have is identify the control strain. So if the researchers were investigating what would happen if they didn't have these genes, our control is going to, of course, be strain 1. Because strain 1 is going to have all of our genes present, okay? So, of course, we're looking at strain 1 is our control. This says use the means and the confidence intervals in Figure 1 to justify the claim that ABC8 is effective at providing resistance. Okay, well, if I look here at ABC8, I can see that the 2 places where I don't have it is in 3 and then also in 5. Notice that in 3 is the only one that's missing, okay? And so you can see that we have these 2 pluses, meaning that it has these 2 genes, but it doesn't have this. So we want to justify that it actually does something to it. Well, you can see that the mean is lower. Well, that's logical, okay? But the thing, in order to be able to say if it's statistically significant, that error bar cannot overlap. So if I look at my error bar for my control, it does not overlap with the error bar from 3. And so that can tell me that the data is statistically significant, and that can help to justify this claim, okay? Now, whenever you have error bars, it's very important that you completely explain which ones you're looking at. The error bars from strain 1 do not overlap with the error bars from strain 3. The ones that is a wild type does not overlap with the one that is has the deleted ABC8. You need to make sure that you're distinctly stating how it's statistically insignificant, or that there is no statistical difference. You have to be very clear about which lines you're talking about. You can't just say the error bars don't overlap. Which error bars don't overlap? And then one more thing also, remember I said that with identification points, you could have just written strain 1, but how hard would it have been to write a sentence that says the control is strain 1, okay? Always make sure that you write a complete sentence to ensure and to make sure that you get that point, because we want to make sure we get our points, right? It's all a points game. And you don't want to not get a point because you didn't write in complete sentences. always, 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 always write every single thing in complete sentence, okay? So again, if you have questions as we go, you're more than welcome to pop them down into the little chat. So part B, there's a lot of verbiage to it. So P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of the cell. CPS encodes an external structural protein located on exocells and that greatly reduces the absorption of insecticides. Based on the information in the data, explain how the deletion of both P450 and ABC8 results in lower survival in bedbugs compared to the deletion of CPS alone. Oh my gosh, that is a lot going on in one question. Like, really? So this is one of those times that why I'm recommending that students take the paper exam. Okay, that was a lot of stuff. You notice that I've underlined all those components because I want to draw attention to those components, right? I want to make sure that I know exactly what I'm looking at. Okay, so I highly recommend that you do the paper exam just so you can write on it and so you can go back to questions, right? The virtual exam, you're not going to be able to go back and you can't manipulate the paper in any way. So definitely take that paper exam. So the question says, explain how I know that the deletion of both P450 and ABC results in lower survival. So if I look here, I have no P450 and I have no ABC8, and then I do have CPS2. So I can see that there has been a decrease in the survivalship. Okay. And now why does it decrease the survivalship? Okay. Why is it decreasing it? Well, we know that ABC8 is going to pump the insecticide out. Okay. So in order to have it has a lower survival because it's unable to pump out the insecticide. It doesn't have 450. It is unable to survive because it cannot detoxify the insecticides. All it can do with CPS is, which is it can just encode this thing to reduce the absorption. So that doesn't do you much good, right? You deleted these, but that's present, so you can't detoxify, you can't pump out, and that just means that there's a lower chance of survival, but it doesn't do you much good, right? And then if we were to look at if we had these two genes and that one missing, what would happen then? Well, now the bedbugs can detoxify and pump out the insecticide, then that causes you to have a higher chance of bedbug survival, okay? Now, this says you get a point per row, and there's 2 points maximum. So I don't know what part of that would have made you think that you had to write both of those lines. Me as a writer, as a student, I'm honest, I probably would have only written one of those. I probably would have written the strain V because that would have shown me how the absence of 450 and ABC8 led to that lower survival. But I think that they wanted you to do each one as if we took these two genes out and then if we took this other one out. So it's kind of a weird question. But that is that question. Again, it's a short FRQ, so there's only 4 points available to it. So our next one that we have is 2017, number 3. So we have gibberellin, and it's a hormone that promotes stem elongation, and then it causes this BACH, that is an enzyme that catalyzes the conversion of the precursor of gibberellin to the active form. So if I don't have GA3H, then I'm not going to be able to have gibberellin, and my stem won't elongate. Okay, so you follow that logic there, right? So mutation in this gene results in a short plant. So as I said before, I can't make the gibberellin, so I can't make the stem elongated, okay? So when a purebred tall plant is crossed with a purebred short plant, all the F1s are tall. Okay, well that tells me that this trait must be a dominant trait, right? And so now we have this F1 is going to be heterozygous. And when the F1 plants are crossed with each other, 75% are tall and 25% are short. So again, that symbolizes there that we have it's heterozygous, a monohybrid cross. One of them is going to be homozygous dominant, 2 of them will be heterozygous, and then the one that we see that's short is going to be homozygous recessive. And then they give us this codon chart. And that was all in the intro to this question. Like, oh my gosh, there's so much information that they've given you. As I said before, you'll want to be able to write on it. So the wild type, I didn't flip the camera, sorry. The wild type encodes this enzyme with alanine of a non-polar amino acid at position 229. The mutant allele encodes this other enzyme with threonine, a polar amino acid at the same position. Describe the effect of the mutation on the enzyme. Okay, well, if you are taking a non-polar amino acid and you substitute it for a polar amino acid, we know that non-polar amino acids naturally are going to fold inside. They're going to fold toward the interior of the protein because they want to hide from the water because nonpolar is hydrophobic. Versus the polar amino acid is going to fold outward toward the aqueous environment. And so when I change this amino acid, it's going to cause the protein to change shape. I'll have a conformational change, change shape, change structure. And when you change the shape, of course, you change the function. Okay. And so it says to describe the effect of mutation on the enzyme, the effect would be you have a change of shape. And that we have, or we could say that there's a change of function. Any of the above would work. Okay. And then I need to provide reasoning how the mutation results in a short plant phenotype and homozygous recessive. Okay. So why, if I have two of these recessive alleles, am I going to end up being short? Okay. And let's remind ourselves that this enzyme's job is to convert it to gibberellin, and gibberellin's job is then to promote the stem elongation. So if I have the mutant, I'm not going to be able to make the gibberellin. It'll either decrease or eliminate the production of that gibberellin, okay? And as you remember, enzymes only speed up a reaction, right? So you don't actually have to have gibberellin there, but it allows this reaction to move faster. And so we found that without the without the enzyme to make that gibberellin, we don't make enough of the gibberellin to allow us to have the super long stem and have that elongation to that stem. And so without it, it's going to, of course, be shorter. So moving on to part B. Using our codon chart provided, predict the codon sequence that results in the substitution of alanine for threonine at position 229. Okay, so literally looking at your chart, you can zoom in. You can say, okay, well, here are my two little sections, right? I substituted something for an alanine to get threonine. Well, you notice that the second and the third nucleotide are the exact same if I look at the first one, second one, third one, and fourth one, right? The only thing that's different is the first nucleotide, okay? So how did I do this? Well, we had a substitution in the first nucleotide. So all you have to do is you can say that the way that we got the mutation is there was a substitution of G to A in the first position of the codon. You could also talk about that we could have had the position of the GC, and then N stands for nucleotide. So you could have had a change of one nucleotide for the other, because that's talking about that it could be any of those four, right? Because you could have had a U, a C, a G, or an A, and it still was the same thing. So we're just saying that we substituted out the G and the A is basically what that's saying. And this last part is looking at the template strand, and it's read the opposite way. So you could either have a C or a T could be switched out because A pairs up with T, and then G pairs up with C. And so if we switch out the T for the C, then that's going to allow us to have that same mutation. I know, a little complicated, but those were the three different possible ones you could have written. I, as a student, would have written out that codon one because that was the most obvious one. I would have said in the codon, there was an adenine instead of a guanine. There was an adenine instead of a guanine. So, last but not least in this question, we're supposed to say to describe how the individuals, and remember, if you have questions, you can pop them down in the chat. Describe how individuals that are heterozygous with one or two copies homozygous of that wild type can have the same phenotype. So why are we able to make a tall plant no matter if I have one of the alleles or two of the alleles? Okay. So as a reminder, this gene is making the enzyme that's going to make gibberellin. Well, in order for me to be the appropriate height, I have to make enough gibberellin. So it's basically saying that if I have one or I have two, I still make enough gibberellin. So one alone produces enough gibberellin. You could also say that the active enzyme produced from one is appropriate. Gibberellin is produced in the presence of one. It doesn't matter. You're just basically saying that you make enough of that gibberellin to get the same height. Because remember, usually when we see recessive traits, it just has to do with that you're not making something. It's the absence of it. And that's why that's why they're recessive, is because the dominant makes something and the recessive doesn't make it. So one recessive produces enough to make a tall plant. Correct. So what this question was saying is you had to make the connection that no matter if you're heterozygous or homozygous dominant, as long as you have the one dominant allele, that one dominant allele makes enough of the gibberellin that would allow you, or makes enough of the GA3H to make enough of the gibberellin to allow the plant to be the appropriate height. Yes. So yes to your question, the student just asked. And last but not least, last question. We can do this. So we have this yeast. It's a single-celled organism. The amino acid synthesis of our yeast cells occurs through metabolic pathways. Enzymes in the synthesis pathway are encoded by different genes. The synthesis of particular amino acids can be prevented by mutation encoding the enzyme. So what they did is they then took two different mutants and they grew it on different medias. You can see treatment 1 has all the amino acids. Treatment 2 has no amino acids. Treatment 3 has all except methionine. And treatment 4 has all except leucine. And you can see which ones have grown where by a plus or a minus. Okay, so we need to identify what is the role of number 1, right? So why do I have this one here that has all the amino acids? It's a control. So the whole reason why we're going to grow it with all the amino acids there is to make sure that the yeast itself is viable, that the wild type, that the mutants are both completely viable, that they will survive no matter what. Because that's making sure that they actually were viable and that they would live in the first place. So that if I find that in one of the later experiments that it didn't survive, I was able to know, okay, well, it was alive in the first place and now it's not. So this treatment must have killed it, is what we're seeing here, okay? So by seeing the positives across the whole row, that's showing us that is our control, and that's a way for us to test that we actually are, that the treatment is not affecting it, that everything's viable, all the good stuff. This whole positive control is just saying that you know what the results are. The results are just making sure that everything works out like you think it will. And I think everything should live because all the amino acids are present. And so that's what a positive control is. So the second part, provide reasoning to explain how mutant 1 can grow in treatment 1, but cannot grow in treatment 3. So if we see here mutant 1, it grew here, but it doesn't grow here. And so we need to figure out why. Well, here, methionine is present, but here methionine is not. So that must mean that mutant 1 is unable to grow if it doesn't have methionine, because mutant 1 cannot produce its own methionine. So methionine used present, mutant 1 cannot synthesize it. Yes, so it's a lack of methionine. Great job. And then last part of this question, which was a common miss by a lot of students. The yeast mate, and they fuse by fusing the 2 haploid cells to make a diploid cell. So basically, mutant 1, mutant 2 are going to fuse together, and it's going to produce this cell over here. Okay. Using the table, predict whether the diploid cells will grow in each of the media. Use a plus sign to indicate growth and a negative to indicate no growth. So here, will it grow? Well, of course, it's our control. It should grow because all of the amino acids are present. Okay. We're going to skip the second one and come back to it. So if we go here, right? So we know the mutant 1 will not grow because it can't make methionine. But notice here that mutant 2 can make methionine and it does grow. So we would expect growth to occur here because of the fact that whatever this one can't make, this one can make. And the same applies here, okay? So this negative shows that mutant 2 cannot live without lysine because it can't make lysine, right? But look, mutant 1 can, which means that mutant 1 can make the lysine for mutant 2. So again, it survives. And if we put all that thoughts together, since 2 can produce what's missing from 1, and 1 can produce what's missing from 2, and they can produce everything else, we would also find that it lives. So, I hope that was helpful. Remember, you are AP Bio Penguins, and you're dressed for success. Good night.